Hey guys, so the purpose of this video is to kind of wrap up the week a little bit and also show you an assignment that you'll be working on next week. Next week is a strange week because we have SAT testing. So our rhythm's going to be interrupted a little bit. So I wanted to make this video to, encapsulate things that are happening this week, but also give you a preview for what's happening next week. This week was awesome. I had a really great time with you guys in class because you were in lab, right? And you were working really hard to find cells in a microscope, two different types. Eukaryotic cells, which came from you, and then prokaryotic cells, which came from a little plate because they're bacteria and that's where they're happy growing. This is actually an image of the cells that one of you took. And somebody in first period took this awesome image. At 40x magnification, right? And there's a few important things that I want to point out and have you guys take a look at. Look at all these features, right? So right here on the outside is a cell membrane, which is made of like a kind of like a fat. It's an oil, really. It's almost like motor oil, as you guys will read. That's sort of the analogy that Bill Bryson makes in chapter 24 of a short history. Calls it kind of like a light motor oil. And then on the inside here, all these dark dots that you guys can see is the nucleus, which is a huge component because it holds some really amazing stuff. That holds your chromosomes, it holds your DNA, right? And what we really want to do next in biotech, after we're done talking about cells this upcoming week, is we want to know what's going on in here, right? We want to be able to start manipulating that stuff. So that's what's coming up here in the next couple of weeks. But look how massive and big and beautiful these cells are. And then you guys got to this, right? And you guys can see like all of these specks, like all of that red. And all of that sea of red is E. coli, right? It's bacteria. So if we even zoom up even further, this is the view at 40x, by the way. Look at all the cells that fit into 40x there compared to there. Right, so these eukaryotic cells are just massive compared to the prokaryotes. But if we zoom in even a little bit, you guys can see like all these little dots. Those are all individual cells. And you notice some of the characteristics are missing from these guys, right? Like these don't have a nucleus. So even if we were able to get like really close, they don't have a nucleus. They have sort of like this rod shape to them. In many ways we could say they're much more simple and some eukaryotic cells are. And that's sort of a feature of eukaryotic cells is they can take on all sorts of different shapes. Getting to the size a little bit more, you guys went through one of these explorations of size and scale this week on your remote days. And here's an example of a skin cell, right? About 30 micrometers across. So much bigger than something like a red blood cell. Back here, you guys can see there's like an X chromosome. And then there's this little tiny like red sliver right here that you guys might be able to see, right? And if we zoom in further, that's a mitochondrion, the powerhouse of the cell. There it is. And then right below that, you can see it's about the same size as a bacterium, and there's specific reasons for that we won't get into right now. But they're almost identical in size, and this is what you looked at in the second day in lab compared to this big giant massive thing. So that scale difference is pretty consistent across eukaryotes and prokaryotes. What's cool about eukaryotes is the different shapes they can take. So this is a good example and will lead us into your task 3 for disease of interest, which is what I want you to work on today. You'll work on task 3. So these are the cells of lactose intolerance. Remember, lactose intolerance is that model disease that we've been talking about in class. And they're really found in the small intestine. That's where lactose intolerance actually makes an impact in the body. And I want you to kind of compare these cells to the last time I talked about cells, which was about cystic fibrosis in the lab primer from last week, from last Friday. Look at the amazing similarities in shape between these two things. And you guys want to notice these little like hair appendages called villi that stick out. And then you can see this little line here, and that's all nuclei sitting there. And you can see the same thing over here, just at a different scale. But really consider this question. What does the shape of these cells suggest about a function they must share? So think about what happens in the small intestine. And then think about what happens in the lungs. And what's the job of those two things? And then how does that relate to the shape is ultimately my question. So here's a closer view of those. Over here, these are your cells in the intestine. You see like these like hair-like appendages. And then over here, you've got these hair-like appendages inside of your lungs. How does that relate to what they do, right? Well, what they both actually do is they take things in. They absorb. So in the small intestine, we absorb small molecules that we've digested from our food. And these hair-like appendages give a lot of surface area. And if there's a lot of surface area, things tend to be absorbed a lot easier, right? Sort of get this idea. Imagine that you guys have a cup of water, right? And you just leave that cup of water out. And then imagine that you have a cup of water that you spill on a table. Which one would evaporate faster? Well, the one that you spilled on the table, because it has a lot of surface area. It's spread out thinner over a larger area. That's what these cells try to do. They try to have a larger area so that absorption can happen much faster. And in the small intestines, we want that to be food and food particles like glucose. And over here in the lungs, we want that to be oxygen, right? We want to absorb that stuff fast. So you get these really similar cells, but there's different disorders that occur here if these things if something's going wrong in these two areas, right? This is just another look, more of a cartoon look at the similarities in cells between these two things. So look at where I've circled here, right? Those are the cells and what they look like in the small intestinal cells. And then look at how incredibly similar they are to lung cells, right? They look almost the exact same, and it's because they perform a very similar function, and that function happens to be absorption. If the shape of these things goes wrong, or if there's a wrong protein in these cells, you can get something like cystic fibrosis, which ultimately comes from a gene, right? So what I'm going to want you guys to do is do something similar, and I want you to examine the cells that your disease of interest actually impacts. So that's task number 3, and that's what I'd like you to work on today, and it's something that you're actually going to present next week. And the format of the presentation is what I'm going to call a 2x2 presentation. I'm going to expect you guys to put together a presentation that will last a total of 2 minutes. And I want you to do that with 2 slides. Okay? So you're going to put together a presentation, and as you're talking, you'll talk for 2 minutes, and you'll have 2 slides in order to do that. And what I want your target to be for this task 3 for disease of interest is what cells are primarily impacted by your disease of interest. Right? Like, what are the actual cells that are impacted? And I showed with you the example of lactose intolerance and cystic fibrosis and the cells that are actually impacted by those diseases. What about your disease? Which cells are they? What do the normal cells of this kind look like? Right? So under normal circumstances in healthy individuals, what do the cells actually look like? And then what about the disease cells? Do they change shape? Is it just a protein? What's going on? So you're going to have to dig and do a little bit of research. You guys should know where to go for some research now. Find what cell or cells are primarily impacted by your disease of interest. Put together 2 slides, and you'll talk for 2 minutes. And the way that's going to look next week is here. So here's our rough schedule. I'll put this up on a module soon. But notice here, this is the weird day. This is Wednesday, where you guys won't actually be in school, and that's going to eat up some time for our red cohort, our A cohort, unfortunately. So here's the pace for the week. On Monday, when I see you guys in person, red group, you'll have time to work on your lab write-ups. I'm going to do a brief kind of set of notes with you, really, really brief. I mean like 10 minutes. And then we'll work on something called cell calculations. And we're just continuing to try to answer the question of what are cells, what do they look like? My gold cohort, you guys will have some questions that you already know what the questions are, right? Something that's interesting that you learned, that you got from that reading, and something that you learned from that reading from Bill Bryson. And you'll have a really short quiz. I'm also going to release cells number 4 that day for you guys to work on. So that's a busy day for you guys. And then we'll just flip-flop on Tuesday, right? So you guys in the red cohort will work on what B did, vice versa. We're getting how that works now. Then Wednesday we have our really weird day. And my recommendations for you guys is that you do these things on that day. You prep for your presentation that's going to be coming up, and you work on cell number 4. And what I mean by responses is you guys are going to be on a discussion board, having a conversation. So you want to follow up with responses. I'll post that stuff in more detail so it makes sense to you as it's coming up. Here, I just wanted to show you what the plan is. On Thursday, right, my red cohort, you guys are going to be doing the cell 4 responses because you're online. And my B cohort, you will be in class together. And what we're going to do that day is you're going to work on your presentations. So for my B cohort, your disease of interest task number 3 is due, and your presentation will be done live in class on Thursday. Okay? For red, we're going to have to do this really differently. I'm actually going to have you guys do your presentations via Zoom. So I'm going to have to teach you guys how to share your screen, and you're going to give your 2-minute presentation right through Zoom on Friday. Then you'll have a lab primer for our next lab, which is going to be DNA extraction. I'm going to teach you guys how to yank DNA out of those cells, those exact cells that you looked at underneath the microscope. Okay, so that's the plan coming up. It's a weird, wonky week and a weird, wonky year. If you guys have any questions about it, make sure that you let me know. I'll make sure to give you guys some really clear communication on Monday so you know exactly what's going on and what to expect. If you have questions for me, just make sure that you ask. I enjoyed the time this week, guys. Let's continue with looking at cells next week, and then we'll get into the good stuff hiding in the nucleus of your cells starting next Friday.